next session of Phylogenetics 101, and we're slowly moving towards creating a tree. So first we had just looking at it. Now we are, what do you use and how should your alignments and your sequences look? And next year in February, we will continue with these sessions, and then we will probably get to models and bootstraps and things like that. And it's all going to end in you being ready to do a phylogenomics course in September next year at our conference. So keep the time. We will let you know about the specific dates and things, but August, September, we're looking forward to having everybody here, maybe in Pretoria for the conference. But enough from me, over to Martin.
Thank you, Finus. Yeah, and thank you for attending. Those of you that are not sitting with us in the room, there's actually just 3 students. Thank you for sitting here. I want to remind you that this is Phylogenetics 101. So we focus really on just basic principles, the basic understanding of some of the concepts, some of the ideas. How do we deal with data? What data should we use? How do we treat the data? So in that regard, I have decided to entitle this session as From Chromatograms to Multi-Sequence Alignments. So we'll just touch on chromatograms very briefly, and then move to multi-sequence alignments. Just want to move forward. Honest, we have a little problem. It's not
Progress.
Yeah.
It's
So if you just bear with me for a moment, we just need to solve this.
Oh, there we go. Screen. Selection screen. Well, problem sorted.
Well,
We're ready to go.
Right. Right. Okay. So I've thought to give you an outline of the steps that are involved in building your data set. So obviously, you will get a DNA sample. That sample might be from blood or from fungi, from mycelium, whatever that might be, bacterial cells or something. You get your sequences from the DNA sequencing facility, and these will be in the form of chromatograms or in the form of FASTA files. And I would want to remind you just to that the FASTA file is actually just not something you can use. It's generated from your chromatogram. You can look at it, but you can't change the sequence. You can't inspect the quality of your base calls. There's very little you can do with it in terms of correcting your sequences. So the first step would be to inspect your chromatograms and make corrections. And these corrections are not just because you think it should be a T or an A, adenine or cytosine. Typically, we will use the forward and the reverse sequences and compare the 2 sequences, and then make corrections based on the base calls in the opposite strands. If you're happy with a chromatogram, you think it's good enough to use, you will align your sequence and then inspect the alignments and make adjustments manually. So some people say you should not make any alignments, adjustments manually, but rather stay with the software that you use, let the software do that, and that's the final product. That is up to you. I typically look at my sequence alignments to check if there's some pathology there or wrong things that are wrong. But that is something you can decide on. I would recommend personally to go back to your sequence, to look at your sequence alignments. If there are stretches that you can see are obvious problems or mistakes, because these software do make mistakes, and I will show you that, then you can just adjust it in whatever sequence alignment editor you use. These editors are things like BioEdit. That's a very popular software for Windows. For Mac, it can be SegL, but there's a whole body of sequence views, and I'm talking about DNA sequence or amino acid sequence views. Right, from there, you can check if there's any unique substitutions in the gaps. Sorry, I just want to correct this, unique substitutions or unique gaps in the alignment. And I will show you examples of these. If there are, you go back to your chromatogram. So if there's anything that looks wrong to you, you go back to your chromatograms and you make the corrections. But again, based on the forward and reverse sequence alignments. Right, so if there's nothing wrong, as far as you can see, you can remove ambiguously aligned regions. That's something that you can do when you have very divergent or variable sequences. One can actually remove the ambiguously aligned regions. That's typically with protein sequences from diverse or very distant taxa, where you get these areas where there's not absolute surety in terms of how strong or how confident you can be in that alignment position. So after that, you can generate a quick phylogenetic tree, and that's something you can do with neighbor joining. You can use a program like Mega. That is one of the programs that I typically use just to get a quick overview. Then that is really important, the next step. So if there's any sequences with unexpected long branches, you have to go back to your sequence alignment and see what's going on in the alignment. It might be that you have to go back to your chromatogram and make corrections. If nothing looks, if the branches are not unexpectedly long, we can look at whether it's some taxa group in a place that was not expected. So you expected to group within a certain family or genus, but now it groups out of that genus. There's something wrong there. If you know that it should be, say, a Agaricus, but now it groups with Coprinopsis instead of Agaricus. You can go back to your sequence alignment and check whether there is not something that you did wrong. Things that we will talk about is the fact that you can have the wrong orientation of your sequence. So you did alignment with sequences that are in the reverse complement. Or what also can happen is you get primers, and I'm talking to the students, you get primers from your friend. It tells you this is a primer that amplifies translation elongation factor 1 alpha, but something went wrong in the line of sharing and getting primers, and what you actually are amplifying is a beta tubulin gene. So if you align those sequences, you will get very interesting phylogenies. You might think you've discovered a new genus or even a new family, but in fact, it was just because either your sequence alignment was in the wrong orientation, or you sequenced the wrong thing. So be careful in the interpretation. And now I'm focusing a lot on taxonomy and diagnostic, because this is typically where we see this. So someone runs to the supervisor and says, wow, we have a new family. The disappointment when the supervisor look at the alignment and see that it is actually the wrong gene. We know it's the wrong gene because you can do a blast search, or it's in the reverse complement. It's a very disappointing feeling, and some of us have gone through this. Right, okay, so if all is good and you don't have to inspect your chromatograms again or align your sequence and then align and go through this whole process, you have an option whether you want to combine your data sets. So you want to do a multi-locus sequence analysis where you concatenate your DNA sequence data, or you don't want to, you just want to do a phylogeny on the single genes. That then determines the next step. So if you want to concatenate your sequence, we will use different software that can do it for us, or you can just copy paste your FASTA files or your Nexus files, the one gene region or partition, as we call it, underneath the other one. Of course, if we work with phylogenomics, we have thousands of genes, or some taxa, or hundreds of genes. That is not something you can copy paste. We use software to do those concatenations. And then if all went well and you are confident in your alignments and the quality of your alignment, you will generate the final phylogenetic tree. Just a reminder of some of you that have forgotten what a FASTA file format is. I just want to get my laser pointer here. So here are two examples. One is in nucleotide sequence format and the other one amino acid sequences. So the FASTA file starts with a header. The header contains the sequence name. That is followed by the sequence. In the next line after the sequence, you will have another sequence or header, with the sequence name. And that's the same for amino acid sequence data. Today, or currently, many of the software that we use can read FASTA files. Some of them can only read Nexus files. For example, Beast is one example. So if you do a Bayesian analysis using Mr. Bayes, that requires a Nexus file format, which I'm not going to discuss. But many of the other software, you can input a FASTA file and do the analysis. And the same goes for sequence alignment. So the files that we use for sequence alignment is typically a FASTA format. In fact, it's always a FASTA format. Now, if you ignore this chromatogram and you just look at the sequence in the FASTA format, I think you will agree with me that this is a terrible sequence or a terrible result. Now, and I'm showing you this with a very specific reason. I don't know how all the sequencing facilities work, but some do send both the chromatogram and the FASTA file. And what I've seen, and I'm not criticizing people, but I just draw your attention to this, is that what I've seen is that people take this FASTA file and just use it as is. This will yield a very bad phylogeny. This is really garbage. And when you have garbage as input, you will obviously have garbage as output. Clearly, from the chromatogram, one can see that this is something went really wrong. You have nice sequences in the beginning, and then you have a lot of ends, which means that the sequencer could not, in terms of the base calling, it could not distinguish between the ACTGs and so on. This, in contrast, is a much better sequence chromatogram, so a much better sequence result from the sequencer. You see most of the peaks are very clear, but there are some ends, and these ends are just because the ratio between the two nucleotides is not at a specific or big enough to distinguish between whether, or to make a call whether this is a G or a T, a C or an A, and so on. So if you have the opposite strand, sequences of the opposite strand, it's easy then to correct them if you have a good and strong signal for that nucleotide position in your opposite strand of the sequence. And then you can use programs such as CLC or Genius and other software to make the correction. So this is very important. So this is step one when you start with your sequencing, with your sequence alignment. You must check and inspect your chromatograms. You must make the corrections, and not just call this, say there where we have a star in this end. it looks like a G. You cannot say that because there's no confidence in even in your own calling here. Here it's even worse. Perhaps that's an A because you have a very high peak, but I would rather go back or do a consensus alignment where you take the opposite strand and align it with your forward strand. So your forward strand, and then align your reverse strand with it to make that call. Okay.
Maybe we should just pause here and ask if there's any specific questions that people would like to ask up to now. We've asked that you place it in the chat box, but we can also allow you to just have a chat. So any questions or anything you want to have at this stage?
Yeah, and I forgot actually to say and remind you that this is actually like a discussion. So please, there are no, I'm hesitant to use the word stupid, but there's no stupid questions. They are just inquisitive minds and people that wants to learn.
I don't see anything in the chat yet, so but please feel free to either raise your hand or add something to the chat, because you know this is, as you say, a discussion and we can interact and talk about it. So everything clear? Okay, let's go on.
Honest, I've just finished my lecturing and my exam, so is everything clear? Nobody asked a question.
Yeah.
Right, okay, so now we can move to multiple sequence alignments, and I'm just going to give you some basic theory. There's a lot of papers that you can read, there's a lot of textbooks. I forgot actually to bring one example that I would recommend, and it's not promoting it, but it's very useful, is the Phylogenetic Handbook. If you just Google it, you will find it. It's every chapter deals with a part or concept in phylogenetics, and after the theory is a practical session in the textbook. Okay, so multiple sequence alignments, that's the foundation of a molecular phylogenetic analysis. That is really the basic. We can do phylogenetics on morphological or phenotypic characters, but in terms of molecular analysis, you need a sequence alignment. Of course, when it's phenotypic data, you have your phenotypic characters and you have your character states for that specific character in a column. Here we align our amino acids or our nucleotides such that all of them are in a column that is thought to be homologous. If we have the same amino acid or nucleotide character, then that position is conserved. But if it's different, then it's derived from the same ancestral state. Okay, so what we're trying to do is to align it so column 1 will be a character. Anything in that column will be assumed as being the result of homology through common ancestry. If there's no character that can be inserted there, we will use a hyphen, which acts as a placeholder. Some of you are familiar with morphological data. If the character is eye color and the character states is blue and green, but there's no eye for some organism, but the color is the character state for that will be a question mark because we don't know. It's the same principles. We also use gaps, and here we can apply gap penalties for the creation of a gap, for the extension of a gap, and we can decrease gap penalties to allow for more gaps and thus fewer mistakes. So these penalties, typically in most cases, we use the default weights because these are typically the best weights assigned through simulations and testing on data sets. So if you know something about the evolution of a specific site, you might change the gap penalties or the weight for making a gap. So first we create a gap, then we extend a gap if there are more than one gap. But in a typical situation, we keep and use the defaults. So if we increase the gap penalty, this may result in missing matches that do reflect homology. We can also use weight matrices, for example, DNA matches versus mismatches, transitions versus transversions. And in terms of protein sequence data, it's the same. We can use mismatches versus mismatches. We can use the chemistry, whether it's a neutral amino acid, what is the polarity of the amino acid. And for these, we apply different matrices. And these can be PAM, Blossom, or Garnet. So there are many of these matrices. They are built into the sequence alignment software. You can select the ones that you want to use, or you can use the default setting. When you use the default setting, you can always go back and do an alignment with a different setting, and then compare your sequence alignment and look for whether, and determine then whether the one is actually to change the setting, whether it's actually optimizing your alignment or making or decreasing the quality of your alignment. For those of you that just want to know how do we calculate these distances in terms of alignment, so all these multiple sequence alignment use distance analysis. There are some that don't use it. They use a Markov or other methods, but in its most basic form, we will calculate first the distance between two sequences. So you have sequence 1 and sequence 2. It's aligned. You realign it in a different way, and you can then determine the distance between these two alignments and make a decision based on the distance, which of the two alignments would be the better one. I will show you in the next slide what I mean. Okay, so min is just the minimum value among all possible alignments. W1 is a mismatch penalty. W2 is a gap penalty. Beta is the number of mismatched pairs, so that will be substitutions. And then gamma is the number of gaps, irrespective of the length. So it's just the number of gaps. Right, here's an example. I've put the equation here just to remind you and what our assumptions are. In the first alignment, we have 1 to 3 gaps, and we have a substitution T to A or A to T. Doesn't matter which direction. The point is there is one substitution. I'm showing it with a star or asterisk, and the others with vertical lines. Okay, we can change the sequence alignment now so that we have a different alignment, still the same sequence, but instead of 3 gaps, we only have 1 gap, but 2 substitution. If you then calculate this based on this equation here, and I'm not going to go into the details of multiple sequences, it's just an example of 2 sequences, you will see that the E value is, or the distance then, will be 13 for sequence alignment 1, while it is 6 for sequence alignment 2. Where do we get 6? Well, we have 2 substitutions with a weight of 1, and we have 1 gap, so with a weight of 4, so 4 times 1 plus 1 times 2, that's 6. Here we have the 3 gaps with a weight of 4 and 1 substitution with a weight of 1, and you can calculate that, and that will get you to 13. So which of these 2 sequences are the best? Anyone? I'm looking at some of the students here. They're too shy. Okay. So E2 is less than E1, which means this alignment will be the better alignment between the 2 sequences. And that's the basis, that's the basic. That is how we calculate the distances. Of course, in multiple sequence alignment, you will use the sum of the weighted pairs. That is a very complex and long calculation, which I cannot explain and do here in this session because we don't have time for that.
before you go on, there's a question from Hendrik, and he's saying that recently he had sequences where both the forward and reverse strands showed that the couple of nucleotides at N because of equal likely bases at these positions. Is it best to leave these as N's, or is it better to use ambiguous IUPAC codes for the two possible bases that could be, as they could represent heterozygous babies?
Yeah, that is a good question. Thank you, Hendrik. If you don't have confidence in the call, you will leave it as N. So if you forward and you reverse, you cannot then make a call based on the peak height and the peak height. So it's going to be an ambiguous site. If you have then in one, it calls a T and the other one a C, it tells me there's something interesting going on there. You can use a UPAC code for that. You can actually tell the program, depending on which software you use, to code it using the UPAC. So you can tell the program to leave those regions where it's ambiguous as ends, or to make the call using the UPAC code. The reason why I would rather leave it is because you don't know, unless you go further in and investigate it. It might be that you have an allele difference there, depending on the ploidy of your organism. But yeah, so if I cannot make a call confidently, I will just leave it as N. And that's not, in most cases, it's not a big problem for your phylogenetic analysis. There are, most of our maximum likelihood analysis can cope with that, and definitely Bayesian analysis. I will just leave it.
There's another question about alignment number 2, where it says both alignments are incorrect for A to a T and a C to a G, but that could happen.
I don't follow why it's wrong.
A can change into a T. It can actually change into anything.
Oh yes, yeah. No, you're absolutely right. Yeah, no, that's, and thank you. Yes. It is just to demonstrate to you how it's calculated. It's not necessarily the biologically correct alignment. So yes, no, you're perfectly fine. You will obviously, if you move this to here, then you will have a gap on that side, which is also possible. But then you will also increase the alignment distance. But just to remind you, this is just to show you how it's calculated. You will see a lot of alignments a little bit later, and I have to move up, speed up. Right, okay, so progressive sequence alignment. It's not possible to do exhaustive alignments. I've put here more than 12 sequences, but that really depends on the computer that you use or the server. Typically, if you have many taxa and a lot of sequences, it will become a very exhaustive analysis and computationally intensive analysis to do the alignments. So most of the alignment programs use a heuristic approach, like Cluster W, where we construct a crude phylogenetic tree based on simple pairwise sequence comparisons. The most similar sequences are aligned first, and then we have a progressive alignment that's controlled by once a gap, always a gap. But I want to remind you that this is not true for all alignments. So if you don't have an iterative method, this is true. So once two sequences are aligned, or a gap has been introduced, it will stay there. Some of these alignment programs can make errors, and this can then be corrected by I. So many people use Cluster X, Cluster W. The newer version of Cluster is Cluster Omega. All of these programs have their advantages and disadvantages. I will show you just a snapshot. So in Cluster X, that creates a multi-sequence alignment in 5 stages. The first is to align each sequence to every other sequence in a series of pairwise alignments. Then a genetic distance is calculated for each pair. I've already showed you how that is done. Then a guide tree is created from these distances, and the guide tree is then used to align the adjacent sequences blocks. And then the sequence blocks are aligned. MUFT and muscle are the two programs that are most frequently used. And this is the reason for that. It is because it uses iterations. So it doesn't stop. Once it has an alignment, it doesn't stop. It breaks these blocks up and realign it, break it up and realign it. So when you do a MUFT alignment using MUFT, MUFT stands for multi-sequence alignment using fast Fourier transform. Muscle has I can't remember the name, but never mind. You can read it up, I'm sure. They both break these sequences up and then realign it so that you get to the optimal alignment. So if you do alignment in MUFT, typically we specify how many iterations should be used in your alignment. Okay, so here's an example that one of my colleagues, Dr. Jane Wright, shared with me.
Sorry, there's a question. It says, I've noticed that MUFT sometimes introduces random gaps. Any idea what could be causing this?
Yeah, so I will actually get to that, but I can tell you now. So when you have sequence, if you have sequence segments or nucleotides that are like microsats that are in, what do you call it, not duplicated, but in tandem. So you have repetition, of sequences. Then it's very difficult for this program to decide where should it be placed. So if you have TA, 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 and in say 6 TAs in the second sequence, only 5 of these repeat units, then it can put a TA on this side, can put a TA on this side, break it up, so that becomes a problem. I am showing you an example.
Okay, thank you.
Right. Okay, so as I said, here's an example of what cluster X did with sequences. That was translation elongation factors from different organisms. You can note, and on this side we have MUFT, which is an iterative process. And you can see, I hope you can see, one can see the differences in the sequence alignment. So we have a block there, that's the same as in MUFT. And then we have, and then we have this positions here. So now we're introducing what I would call extra evolutionary steps in terms of you have to open a gap and then you have to add a gap or gaps. So you have to open the sequence and include gaps with cluster. Whereas in MUFT, here this makes more sense to me in terms of in terms of biology. So intuitively, it makes more sense because here you have a large indel. An indel refers to insert and deletion versus the many indels here that you will create with cluster X. Here's another example, and perhaps that can also be an example for the question that was asked. Clearly, you can see this is just a random placement of CCA. There's some homology, but there's no confidence in this placement. Rather, there's perhaps more confidence in putting it on this side, and that was discovered through the iterative process that MUFT used. So cluster this in the first steps, it aligned the two sequences, and this was the result, and it stayed like that. In MUFT, it might have aligned it and got to this result, but through iteration, it optimized the alignment so that we only have one big indel here instead of these different indels. So here we will have an indel, then an insertion, and an indel, an insert, and excuse me, I want to correct myself. So we will have a deletion an insertion for that sequence, a deletion. Whereas here we just have basically one evolution step, and that was the complete deletion of that region. You can then, of course, go back and check whether it's codons, what that was excluded, doesn't make sense. That is just the downstream analysis to ensure that your sequences are optimally aligned and doesn't make biological sense. Right, to make your life very complex, there's at least 103 multiple sequence alignment programs that one can use. And I will refer you to this website. If Melandre, that's assisting us here in the room, if she can't get to this URL in time to post it, I will share it with her, and she will then post it on the WhatsApp group for those that are members of the WhatsApp group. So back to this list. All of these, some of them are a bit archaic, like cluster X, which is listed here. Each of them have different approaches. It could be some difference in, like I've already explained, like MAFT that has an iteration process. In general, and I say this very, very carefully, because I don't want to say that this is a product software you must use or not use. It really depends on the data, the data sets, the complexity of your data. But in general, most people use MAFT or muscle because they yield optimum alignments, but they also can deal with many sequences. In my own personal research, when we do phylogenomics, we use a lot of sequence data. I prefer to use MAFT. All of these, both of these programs, muscle and MAFT, can be used in the command line. So you can run it on your own computer, or you can run it on a server, which make it, of course, very easy then to use when you have large data sets. This is just a study that was done to look at different newer type of software. You can still see MAFT is there, muscle is there, but among all of these, MAFT and muscle performed really well. These programs have differences in terms of how accurate they are, how fast they are here, and then their sensitivity. So, but if you look at this table, I don't want to go and spend a lot of time in here, but both MAFT and muscle are dealing well with large data sets and complex alignments. Sometimes a program like ProCons or Satay might yield better alignments, but that really depends on your data. Okay, so now we take a step into the direction where you have your sequence alignment, and for now, I'm just going to remove ambiguously aligned regions. We will deal with that at the end. Okay, now you have alignment. What do you do with your alignment? You have to go to your alignment and then make sure that the alignment is actually the best possible alignment that you can get, that there are no errors in your alignment, obvious errors. And a very easy way to do this is to generate a phylogenetic tree using neighbor joining, because you will immediately see if there's some, let's call it pathology, in your alignment. Okay, so if there's any sequences with unexpected long branches, you go back to your sequence alignment, you go back to your chromatic graphs, you inspect them, inspect your alignments. If there are taxa that are not grouping as expected, if it's yes, you go back to your chromatic graphs, grams, and your alignment. So let's deal with those. So here in this box here, I have a phylogenetic tree that is the true tree, the correct phylogeny. Why do I know it's the correct phylogeny? Because, well, I've done the analysis, but my examples here were made by changing the sequences to demonstrate it to you. Okay, so in this phylogeny here at the top, we see that sample 3, 4, 5, 6, they group with a species called Neophysicoccum parvum. All of these are fungi. They are actually endophytes of trees. Sample 1 and 2 belongs to the species Lasidiophoda, Gonobensis. Okay, so we know this is the true tree. And if you wonder why there are no bootstrap values, it's just to simplify it visually so that you can see what I'm talking about. So here we have two things that happen. I actually should have separated them, but let's deal with them now. So on this tree, we have a very long branch, okay? So when we see that, there are various reasons that can explain this observation. It might be that this is a different species. That might be true. Or there might be something wrong with your sequence. So there might be sequence errors. Or it might be that this lineage, for some reason, just evolved really fast. That can be true. But in general, when we see this, I suspect that there is something wrong with your sequence. So something wrong, going back to your chromatogram to inspect it to see if your base pools are right. On this side, we have something that's really, really, really weird. Okay. These samples are all part of this family of fungi. Yet, they group completely out of the family. In fact, they group completely away from the outgroup. So here you can see the outgroup, Melanops Tularsinia. So something is completely wrong here. So either you've made an amazing discovery, it's a completely new family, or something is wrong. So how are we going to do with this? Okay, first let us look at branch lengths. If you go back to your sequence alignment, you will notice that there are mutations, but it's not true mutations. It's missing calls, wrong base calls. For example, all of these have either a G or an A. Now suddenly you have a T. And I can tell you this T comes from the fact that when you run your sequence on the capillary, there was overflow. So when you have T, sometimes it calls more T's or less T's. That really depends on the chemistry. But many times we have T, and you will find that there's another peak for a T. And the program read it as a T. But it's not a real T. It's just because there was overflow during the migration, or the movement of your nucleotides. Right. Okay. So here's another example. All of them, except for the outgroup, has a G. And now we have, I hope you can see what I'm showing. Now, suddenly there, I've lost my place. Let's take a different one. All of them here are an A. Suddenly we have C there. So that is obviously wrong. Now, I would not just change it. I will go back to my chromatogram and check and make sure what's going on. And you will probably see that there's actually more T's called in the forward direction or the reverse direction, and you can fix that. So that is then really easy to fix. You cannot fix really anything that's deposited on GenBank. That's there, it's there. I would not touch it. That's my personal opinion. If you differ from opinion, that is your prerogative, but I would not. Right, so the possible reasons for this is wrong base calling. So you can correct your sequences using forward or reverse sequencing, or extremely fast evolving lineages. And if you really want to, then you need to, and you really want to understand what's going on there, you increase your sampling size. So what happens then, if you have a large sampling size and there are more of these samples that belongs to this group, you will form a clade or a group. And then if there's good bootstrap support, statistical support, then that's fine. You leave it. Okay, here's just an example, that same example that I've discussed. Okay, here we have that fantastic discovery that you've made. And I can tell you, most of the time when I see this, I know something is wrong with the orientation of your sequence, or you have sequenced the wrong gene. Typically, it's the orientation of the sequence. Most people know they get the right primers. Okay. So the possible solutions, possible reasons, sequence in the reverse complement. So just reverse complement and realign to solve this problem. Non-homologous sequences are used, that's a possible reason, and use then the correct gene. So exclude that, do the sequencing again and include the correct region. An easy way to see if you have sequences in the incorrect orientation is to use MUFT. So MUFT has a nice function that shows you whether sequences might be in the incorrect orientation. So this is the landing page for the online version of MUFT. You place your sequences in here. I've done an alignment, and what I see are these blue lines. When you see these blue lines in these graphs, you immediately know, okay, I have sequences that are in the wrong orientation. You go back and you do your sequence alignment. You can make you reverse complement it, and then of course you have to realign your sequences. A nice way of dealing with this is in MUFT, you can also tell the program or select the option where the direction of the nucleotide sequences can be adjusted according to the first sequence. So if you check this box or this radio button, then you will get as an output, it still tells you that there are sequences in the reverse complement. But in this case, because you've checked the orientation option, now the program has reverse complemented the sequence and aligned the sequences. And here you can see this is a much better, it is the correct alignment. And I will be happy with this. It also gives you an R in front to tell you which sequences were in the incorrect orientation. I think there was a question, so I will just stop there.
No, there were just two links that Milan Grace posted for us.
Great, thank you.
So the one for all the tools and the one for math. So if you want to get to it, the links are there. So thank you for that.
Thank you, Melandre. And by the way, so I give you the background here and I show you the results and so on. But the students have also a group where they do this online using the software. So you're welcome to join them. So it's not just, it's students in the sense of students learning, people learning. So you're welcome to join the WhatsApp group. They have meetings. I don't know how frequently, but where this is then demonstrated and people are helped in solving problems. And if there are problems that cannot be solved, we can help the group by offering our help as more experienced users. Okay, now we get to the point where you are happy with your alignment. There's no errors. The next step would be you can do this or you don't have to do it. It depends on the variation in your sequences. So many people, when we do alignments of protein sequences, amino acid sequence, often those alignments are really difficult to do, and you get many ambiguously aligned sites. Here's just a DNA sequence alignment where I show the effect of a repeat nucleotide. So on this side, we have CCC, and I have an alignment. I've aligned here A under C, C under that C, but you can also have it on the opposite side, on the downstream side. You can put AC on this side. It makes no difference. So in a way, this is just an aesthetical alignment. It doesn't change anything, but in terms of the phylogeny. But we get worse cases where it really doesn't make sense. Here's an alignment with two different programs, and what I'm showing you here is that firstly the effect of two different software, but also how difficult it might. Showing you the effect on repeats on different software. So here we have, I think the bottom is muffed, the top is muscle, will be worse with cluster, but it really doesn't know where to put these, how to align these repeated sequences. Now you can leave it like that. I recommend, if you have really diverse sequences, that you exclude these gaps. One can do that, or you can use software to determine what's the confidence in your alignment position. And there are different software that you can use: ClipKit, BGME, TrimL, Gblocks. Each of them have different principles in terms of how they decide on whether to exclude or assign a confidence value to that nucleotide. If you are dealing with sequence phylogenomics, many people will be familiar with TrimL. But I will show you the result from BMGE. It was published in 2010, but it's still used frequently by the community. So BGME stands for Block Mapping and Gathering with Entropy. I'm showing the reference for this paper here, and you can read more about it. Okay, so this is the landing page for the website. The other software you will have to download on your computer and install it. They all run from the command line. The reason why I'm showing you BGME is not because I have a preference for this, but in terms of just demonstration, it's much easier. So here I have the sequence, it's already aligned. Now we want to exclude the sites with low alignment confidence. If you use sequences from amino acid of proteins, so amino acid sequence alignment, it will then use a blossom or whatever matrix is then used for proteins, and for DNA, it's a PAM matrix. There's not time to go into these matrices. Right, okay, it's going to auto-detect the alignment. It will see that it's DNA. And this is the result. So what it does is then to remove those positions in the alignment where there's low confidence in terms of the algorithm that's used. Now you can't see much here, so in the next slide, I'm showing you, here's showing you the result. The top is directly from MUFT, and the bottom one is the analysis using BMGE based on the alignment from MUFT. Right, okay. So as an anchor point, I've used this repeat region. This is a very constant region, so it didn't have any problems there. But if you closely inspect the sequence alignment in the top, in comparison to the alignment in the bottom, you will notice that some regions are still there, but some regions have been excluded. So this is the intergenic spacer region. It's not so, actually not so diverse. In new amino acid sequence data, when you work across orders, families, you get very divergent sequences. And this is a perfect way then to remove those sequences. And typically that's what we will do in a phylogenomics analysis. Okay. Good. Last part is just the decision whether you want to concatenate sequence data or not. You can generate gene trees. So then, of course, you will not concatenate your sequence, but when you do a multi-locus analysis, you want to concatenate the data so you have one data set that is then used in your phylogenetic analysis. And for this, you can use false concave G. For this, you will need PUL. PUL must be installed on your computer, and then you simply run the program from the folder where you have your data. So you will put your data in the same folder as the program and then run it from there. So first of all, the sequences must be aligned. You can't do a concatenation on unaligned sequences. That will not work, okay? Then the sequences can be in FASTA format, Phylop, or Nexus. Most of the time, we just use the FASTA format. In fact, I take FASTA format from beginning to end in most of the software that we use. Mr. Bayes, if that is the Bayesian analysis program that you want to use, then of course you will use a Nexus format. The sequence names must be identical. So if you want to go back here, if the first sequence is CMW, that's the number 1 _ ZIM, then in here there must be another sequence with exactly the same sequence. So it's search through your data set and find sequences with the same name. So those sequences will then be linked. So sequence 1 is, say, ZIM2, and your second gene, sequence 1 is ZIM3, but the next sequence is ZIM1. It will know that that ZIM1 is then belonging to ZIM1 in the first sequence or data block. You do not need a complete sequence for this. We can, what we call, produce a Swiss cheese data set. Many times when we do sequencing, sometimes we just cannot get a sequence for a gene, for a specific sample. And that is just because there are many reasons for that. So in that case, you can do a Swiss cheese, but I recommend this only when you have larger data sets. If you only have 2 genes, this is going to complicate things. As I said, it's a command line program. There's the URL if you want to download it, and you must have PUL or PUL Strawberry on your machine. Okay, I'm not going to go through all of this. And the reason for that being that we're already at 5 o'clock and we need to wrap up. Just to say that if you open the program, you will have this interface. You control the options by selecting the key for that. So if I want to change the Nexus file to Mr. Bayes, I press on N and enter here in the command line, and that will then change the setting. So if you want to change settings, you just use these keys on your keyboard. Okay, press the key, press enter, and it will change to a different setting. From there, the last step will be to press S, enter, and it will start a run, and output files will be produced. These output files will be in the FASTA format, so it's concatenated. You will have an Excel spreadsheet that shows you what the partitions are, how many genes there are, what are the transitions, transversions, and so on. And you can, if you selected a Nexus file, you will also have a Nexus file. Okay, so we're getting close to the end. If you are very happy with everything, you're confident in your alignment, then you can generate your final phylogenetic tree, which is not part, it's not a discussion that's part of this session. Now, I just want to show you something else that you can do. And that is when we have very divergent, so really variable sequences, but it's coding sequence. We can take our DNA, we can translate it into amino acid sequence, align it, and then back translate the DNA, because by doing an alignment on amino acid sequence is much easier, gives you much better alignment because of the redundancy of the code. And then this will yield a much better alignment with higher confidence. Talking about the DNA again, after you've back translated it. So there are two programs that you can use. There might be others. These are the ones that I use. The one is RefTrans, and the one is Translator X. And I would love to show you Translator X online, but unfortunately, because of the X and the translator on this website and this IP, because of this address, I can't remember what is this. IP address, our university blocks it. So it's either because it's Translator X or it's this. But never mind, I have a screenshot for you. Here's the sequence alignment using MUFT in RefTrans. You can see it's really, really easy, straightforward. It's a very nice alignment. You have gaps here, which is good. Then, if we back translate it, we have an optimized alignment, DNA alignment, based on the amino acid, which then serves as the backbone for our DNA sequence alignment. So that's in RefTrans. An alternative is Translator X. What's nice about Translator X, it shows you the codons in color, so you can very quickly see where there are codon differences. These might be of very great value to you. You can also see where there are deletions, and one can see that there are 3 dots here or 3 gaps. Of course, that's a codon that was deleted in this organism. So visually, it's really nice software. You can very quickly see what the differences and does it make sense, so what the differences are, and does it make sense to you? You can, of course, then download both programs in RefTrans and Translator X, the sequences in FASTA format. So just some final thoughts and recommendations. So these are my final thoughts and recommendations. There might be many other final thoughts and recommendations from our audience, things that they can inform you about, some pointers and so on. So first of all, I would say in our era, get familiar with the command line of your operating system. We are moving towards a place in time where if you're not familiar with the command line, you will struggle, because especially when we want to work with large data sets. Many of this analysis that's conducted with in the field of phylogenomics are done on a server, and that works on that uses the command line. So in Windows, it would be the command prompt. In Linux, it would be the command line. Or terminal, and on Macs, it will be the terminal. Reader manuals, there's no way that we can provide enough training so that you fully understand all the complexities and nuances and all the aspects of this different software that one can use. So please, my suggestion to all my students, and that includes my own students, is please read a manual that will solve so many problems. Inspect and correct your chromatograms, fundamental step. Interact with the alignment results and the associated phylogenetic tree generated from the data. It's a two-way street. You take your phylogeny, does it make sense? You go back to the alignment, and so you go forward and backwards. In my opinion, phylogenetics is the same as in the lab. We are doing experiments until we find the optimum and the best result. So whether you do things in bioinformatics realm, it's like a lab. My computer is my lab. Okay. Be sure that the multi-sequence alignment makes biological sense. Compare like with like. You cannot compare beta tubulin with translation elongation factor 1 alpha in the same sequence. You can concatenate data, of course, but you cannot align in a one data set Same, two different genes. Remember, garbage in, garbage out. If the homology of a region is in doubt, please throw it out. Manually edit alignments if needed. You really have to go back to your alignments and just check. Even if you do phylogenomics, once you've concatenated your sequences, you can scroll through your thousands of nucleotides and check whether there are regions that doesn't make sense. And believe me, you will find some pathological sites. Okay. And then remove ambiguously aligned regions. You can trim the ends, so the beginning and ends where you have only gaps, and replace it with ends or X for amino acid sequence, N for nucleotides. And then try to avoid the most divergent regions of a molecule. But that really depends on your own questions and what you want to do. I think this is my last slide. Thank you for joining us.
Thank you, Martin. There's a question from Bianca, and it says, for these variable sequences, if you are not confident in the introns that are predicted by the annotations, how do you deal with that, since you said you need coding sequences?
Well, Bianca, you can't, of course, you can't use introns because you use coding. The same as you, so for people working with ITS, the internally transcribed spacer region, you deal with it in the same way that we deal with these markers. If you have very divergent taxa, so taxa that are very distant to each other, you will have problems in your alignment, but that's true for your markers as well. So if we take ITS and we do use that to generate a phylogeny over families, you are going to struggle to get proper alignments. So in that case, we will not use ITS. And that same goes with your introns. If you're not confident in your alignment, and I didn't talk about alignment scores and looking at different alignments and use different methods to determine which is the better one. If you're not confident in it, and you are actually asking this in the context, I think, of phylogenomics. Typically, you will have many more genes to use, unless you are interested in that very specific intron, the phylogeny of that intron and how it relates to the phylogeny of the species. But that really depends on what your question is.
Okay, I haven't seen any more questions, so I think it's time for us to say goodbye. There's a song like that.
It's time to say goodbye.
So, time to say goodbye. So thank you, Martin. Thank you very much for your time. I think it was, again, a very useful session. And we are really very happy that you are willing to help us and go through these sessions with us. We will try and get it posted as soon as possible for those that have missed it. And if you've missed our first session, you're welcome to have a look on the YouTube channel, the SASSB one, and watch this space. We'll be back in February when everybody is well rested and ready to tackle new challenges for the next year. We're looking forward to linking up with you again. And thank you very much.
Also from me, thank you very much, and have a very good holidays and festive season.